Cool. Yeah, so I'm Eric. I'll talk about some work we did in collaboration with Roma, James Zoe on building this model SPRITE that to improve spatial gene expression predictions using information from gene and cell networks. So first, a quick overview of kind of spatial and single-cell transcriptomics. Single-cell transcriptomics at the dissociated level is a very common technique nowadays. But basically, in summary, you would take a tissue, you would dissociate it into either single nuclei or single cells, lyse those cells, and then recover the RNA, copy it into copied DNA, and then sequence that. And then recently, a more kind of exciting approach to retain kind of the spatial nature of the tissue has been developed in kind of these imaging-based spatial transcriptomics methods, which use these kind of hybridized RNA transcripts to target a panel of different RNA species in the tissue. And then you do sequential kind of fluorophore imaging to decode unique RNA transcripts and their location in two-dimensional space. And so what do these two types of different data modalities look like? First, for single-cell RNA-seq, you roughly get whole transcriptome coverage, so around 20,000 genes, for anywhere between a few thousand to maybe tens of thousands of cells, depending on the amount of input. On the other hand, you have a kind of different dimensionality for the spatial transcriptomics for imaging-based technologies, where you can get maybe an order of magnitude more cells than single-cell RNA-seq, so maybe in the order of tens of thousands to up to 100,000 cells for a given sample. But generally, you're limited to less than whole transcriptome level coverage. So usually, you pre-select a panel of genes somewhere maybe in tens, hundreds, maybe up to 1,000 genes for each experiment. So this observation kind of begs the question, can we get the best of both worlds? Can we both get the whole transcriptome coverage of single-cell RNA-seq and then also get the spatial information that's captured by these imaging-based technologies and the order of magnitude greater number of cells in that case? And a few methods have been developed for kind of addressing this task. First, people have taken single-cell RNA-seq and try to transport it onto the spatial dimension. So you map each single cell in the single-cell RNA-seq data set to a reference kind of XY coordinate in a spatial data set. And then conversely, people have also taken this opposite approach where you do imputation, where you take the spatial data set, you look at all the genes that aren't covered as these like missing genes, and then you impute the expression of these missing genes using a reference single-cell RNA-seq data set. And this will be kind of the approach I'll be talking about a bit more throughout the rest of this presentation. So there's been a lot of methods proposed for this imputation task. For example, a recent benchmarking paper looked at 8 different methods for across a variety of different contexts for imputing basically this spatial gene expression. Zooming in on kind of one of these methods, which is kind of emblematic of a lot of different methods in this kind of class. You would take your single-cell RNA-seq data set, your spatial data set, you do some kind of joint embedding to class them into a shared embedding space, and then from that space you can map kind of nearest neighbors. So for a given spatial cell, you can look at the nearest neighbors in kind of the single-cell RNA-seq space, and then use that to kind of aggregate information and impute the rest of the missing genes in the whole transcriptome of that cell. However, kind of many of these methods don't use any relational information. As you can see from this example, they generally just take these tabular matrices of genes and cell counts. And instead of using any relational information that's like missing, they kind of just do this kind of very, very basic joint embedding. And so kind of the thrust for this project was to ask if we can build in this relational information for any type of kind of black box imputation. So first you have gene co-expression relations. For instance, genes that are correlated in space or part of the same gene module might have similar prediction errors, and sharing that information during prediction could maybe improve those predictions. We encode this in this reinforced step in the SPRITE algorithm I'll talk about later. And then maybe even more importantly is this idea of a spatial proximity relationship. So cells who share similar transcriptomes but are also located in close proximity in space probably have the most similar types of gene expression. And so how can we leverage that information that is kind of ignored in a lot of these current models to improve the predictions of spatial gene expression? And so to introduce kind of our algorithm for modeling these two types of information, you start with kind of this like spatial transcriptomes data set, so cells in two-dimensional space. You do any of these like black box gene imputation methods. You get an imputed gene expression for a missing gene that wasn't in the original data set, for instance, gene J here. And then you can take that as input into our method, which is kind of a post-processing kind of meta algorithm. It takes this black box imputation, does a reinforced step. We build this gene correlation network and make predictions of both seen and unseen genes to then calibrate kind of predictions across this gene relational information network. We get these kind of post-reinforced expression And then we use that as input into the second step of the algorithm, which is to smooth it across kind of the spatial dimension. So for cells that share both similar transcriptomes at the measured level, we then smooth over similarities in both proximity and the gene expression for these unseen genes. And then we get this kind of a SPRITE predicted, smooth and reinforced spatial gene expression matrix as our output, and then evaluate that compared to the baseline prediction on various downstream tasks like clustering, visualization, cell type classification. So first, to give an overview of how well SPRITE performs compared to the baseline kind of black box imputation methods, we see that generally boosts imputation performance. We've looked at benchmarking across 11 different pairs of both single cell and spatial data sets. So here on the left-hand side, I show improvements as measured by the Pearson correlation between predicted and actual expression. On the right-hand side, I show the mean absolute error as another metric for performance quality. So again, we have the dotted line at 0 is basically no improvement over baseline, and anything above that line is an improvement. So generally, you see that across lots of different genes and lots of different data sets and imputation methods, you generally see improvements across most cases, where generally the median or mean kind of improvement is positive over baseline for SPRITE. Another way to visualize this more concretely is to look at, for these different contexts, which is a combination of an imputation method and the single cell or spatial RNA-seq kind of data set pairing, how many of these do you get improvements in at least one of these metrics? So you see across basically all the cases, more than half of them you get improvements in both metrics, and in all of the cases you get improvement in at least one metric over any of these existing baseline imputation methods. And similarly, when you zoom in at the gene level, you see that only for a very small minority of cases do you actually see no improvement across both of these metrics. To give kind of a better intuitive understanding of what's going on, you can look at a few visualizations of this in the spatial dimension. So here is like a visualization of the mouse hippocampus coronal section. You see kind of high expression of this SLC17A7 transcript in the excitatory neurons in the cortex. On the left-hand side is the measured expression. In the middle is kind of this baseline imputation from the SPAGE algorithm. And then on the right-hand side is what we get after we do our SPRITE post-processing, where we rescue kind of this cortical-specific expression of this transcript. And then similarly here, you have a different case where we maybe dampen the overly optimistic predictions from an algorithm, Tangram here in this case, where we see that in the measured expression is actually very selective to this band of cells. But then, from the baseline imputation by Tangram, you see kind of very promiscuous kind of predicted expression across most of the tissue. And then with SPRITE, we kind of dampen it down to this like selective expression across this narrow band of cells in the tissue. So now that we kind of talked about improvements over kind of at least the prediction task, I think the more important question is basically, are these improvements actually important, and can they be carried over to downstream analysis tasks? And so we look at this across a few different methods like cell type identification, cell clustering, data visualization, and training models, and then a few other things. And so these, we basically see that SPRITE does yield pretty good improvements across most of these cases. For instance, data visualization, we compare baseline to SPRITE, and we see that in baseline cases you get pretty messy kind of UMAP visualizations, but in SPRITE you get much better separation of these different layers of the tissue. Across multiple data sets, we see a very similar case where you see both baseline imputed expression for visualization decreases in quality, but then it's rescues like SPRITE to basically the same levels as what you would get by visualization on the measured transcriptomes. And then also we look at cell type classification, where we build these models using either the baseline imputation or the SPRITE predicted expression. And you see across, again, multiple cases, you see improvements for all these different contexts where you train the cell type classifier on the SPRITE predicted expression instead of the original kind of baseline predictions. So in summary, SPRITE is a meta algorithm that reinforces gene correlation information and smoothens kind of spatial relationships. It improves spatial gene expression imputation across most data sets and imputation approaches. And then also boosts kind of some of these downstream visualization tasks like data visualization, clustering, and cell type classification. So again, I want to thank James and Rome for their collaboration on this work, and also to the Brunet Lab and Zoe Lab for being kind of my home at Stanford scientifically, and to some funding sources as well.
Thanks, Eric. We have time for one very quick question.
Thanks. Great talk. Can you elaborate on how SPRITE improves data visualization? I saw the component separation is very good, So can you, yeah.
Yeah, so we evaluate kind of the data visualization using kind of a Spearman correlation between pairwise distances in the original high dimensional space and the low two-dimensional visualization space. And in each of those cases, we take, either the measured gene expression, so the original kind of ground truth, build a visualization of that using the same hyperparameters for each of these visualization cases. And then we do the same thing for either the baseline imputed expression or the SPRITE prediction. And then you can just kind of compare that statistically in terms of all these different combinations of data sets and imputation approaches. And SPRITE generally outperforms the baseline imputation and performs at a similar level to the ground truth visualization.